Hello everyone, welcome to Get My Class. Let's solve this question which says, principle of ELISA is option A, antigen-antigen interaction, option B, antigen-antibody interaction, option C, antigen-antibody neutralization, option D, all of the above. So let us see. The question says principle of ELISA is. So what is ELISA first of all? ELISA is nothing but E for enzyme, L for linked, so it is enzyme linked, I for immunosorbent assay, I for immuno, and S for sorbent, and A for assay. Okay, it is a method to detect for the presence of a particular antigen. Okay, you can detect the presence of a particular antigen, and how can you detect? You can detect, okay, by testing for the presence of antigen, or you can test for the presence of antibody. So if you test for the antibodies, that means that in that particular person, those antigens are present. That is why those antibodies have produced. And directly you can test for the antigens, or indirectly you can test for the antibodies. Okay, but in both the cases, what is the result? Whether the person is having the pathogenic antigen or not. Okay, whether he is infected or not. Okay, so here the particular antibody which is added to detect the antigen, to detect the antigen, it is being linked with enzyme, a particular enzyme, and this acts as a marker. Okay, this acts as a selectable marker, or you can say this acts as a marker. That means you can know whether there is that a particular antigen is present or not, because if these enzyme linked antibody binds to them, then it will produce certain color. Okay, the production of color shows that that particular antigen is present, or that particular antibody is present. Okay, so basically the principle is based on the antigen-antibody interaction. Okay, antigen-antibody interaction. Okay. So let us see. Option A says antigen-antigen interaction. No, it is not antigen-antigen. It is antigen-antibody interaction. Okay. So that is the correct answer. Option B says antigen-antibody neutralization. So we are not talking about neutralization. We are talking about detection. Neutralization is a process where we try to complex this antigen with the antibody, and we try to neutralize the effect of antigen. But here we are not trying to neutralize the effect. We are trying to detect the presence of a particular antigen. Okay, so either directly or indirectly. So option D says all of the above, which is incorrect. So let us conclude. So what is the correct option? The correct option is option B. I hope you understood the explanation. Thank you, everyone.